FSc Biology Part 1, Chapter 4, The Cell, which is most important because of its high weightage in the PPSC lecture and subject specialist Biology and Zoology exam. In the previous part, we have discussed the first 10 topics, while in this video, we will discuss the remaining 10 topics from glycosome to prokaryotic and eukaryotic cell. The most humble request is that please watch the video online and stay tuned till end. Please concentrate and focus in depth on these all topics. Let's study together glycosome. They are found only in plants, means they are the only organelles which are found only in plants. They are abundant in plant seedlings, means the plant which have their growth in from seed, or they are very young. They convert stored fatty acids in seed into energy to begin a new plant. It is very important that a new plant need energy, and this energy is being stored in seeds by glycosome. Glycosylate cycle, very important to remember. Glycosylate cycle convert fatty acids in seed into energy to begin a new plant. Glycosylate cycle convert fatty acids into carbohydrate in glycosomes. Peroxisomes play catabolic and anabolic roles in plants. Both roles are played in plants which are catabolic and anabolic. Castor bean and soybean seeds are rich in lipids, means castor bean and soybeans have lipids. This is very important fact to remember. Glycosome convert fatty acid into succinate in such seed which are rich in lipid like castor bean and soybean. Glycosome are present for short period in lipid rich seeds. They are absent in lipid poor seeds like pea. This is also very important, that which are lipid poor seeds. Pea. Vacuoles. Vacuoles are large and abundant in plant cells and push organelles aside. Large vacuoles are formed by coalescence of smaller ones or the attachment of smaller vacuoles during growth. Vacuoles serve as storage of water and metabolic intermediate, mean that intermediate metabolic are also stored in vacuoles. They prevent dilution of cytosol. They are major contributor to turgor, which provide rigidity to leaves and younger parts of plants. They are single membrane bound, which is called tonoplast in plant. Tonoplast is the membrane which surround vacuole in plant. Cytoskeleton. Cytoskeleton proteins are tubulin, actin, myosin and tropomyosin. These are very important to remember, that the proteins which are found in the cytoskeleton are tubulin, actin, myosin, tropomyosins. Microtubules, which are parts of cytoskeleton, are made up of tubulin protein. This is also have been asked in the previous paper, that microtubules are made up of tubulin protein. Cilia, flagella, basal bodies and centrioles are assemblies of microtubules. Microtubule are assembled with the cilia, flagella, basal bodies and centriole. Amoeboid and cyclosis movements are due to microfilaments. Intermediate filament determines cell shape and integrate compartment. These are two basic functions which intermediate filaments perform: determine cell shape and integrate compartments. Microtubules are long, slender, unbranched tubulin protein structure. They assemble and disassemble spindles in mitosis. Microfilaments are made from actin protein and are more slender. This is also very important, that microfilaments are made up of from actin protein. They help internal cell motion, and intermediate filaments are in between tubules and filaments and maintain shape. Now, centrioles. Centrioles are absent in higher plants. Very important fact to remember, that centrioles are absent in higher plants. Each centriole is consisted of 9 microtubules triplets having 2 centrioles are placed at right angle to each other external to nucleus, means centriole are placed external to nucleus. Centriole play a role in location of burrowing or burying in cytokinesis and form cilia. Thread-like structure are called cilia. Spindle fibers are formed when they move opposite poles during division. Spindle fibers are also thread-like structure which are important during cell division in mitosis and meiosis. Please notice and remember that mitochondria. Mitochondria are powerhouse of eukaryotic cell as they form ATP. Two most important things to remember are they are found in eukaryotic cell and they are powerhouse of eukaryotic cell because they form ATP, the main energy currency of cell. They appear like rod of filaments or vesicles under microscope. They are in the form of filaments or vesicles. It is also very important. They are double membrane bound, outer is smooth and inner has infoldings. Folds in inner membrane which extend in matrix are called cristae. Definition of cristae is also very important, that the folds which are in inner membrane and extend in matrix are called cristae. Mitochondria has their own DNA and ribosomes. They are self-replicating organelles, means they can replicate itself and form copies. Inner layer has small knob-like structure, the F1 particles. Matrix consists of enzymes, coenzymes, salts for Krebs cycle, respiration and metabolism. So the main function of mitochondria are these. Cristae is made up of lipoproteins having different enzymes in it. Cristae is made up of lipoprotein which have different enzymes. F1 particles are embedded in cristae. When cristae rupture or broken, F1 particle come on the surface. F1 particles actually convert ADP adenosine diphosphate into ATP adenosine triphosphate. Mitochondria convert food into ATP. The cells which have high metabolic activity have more number of mitochondria in them. For example, muscle cells, and the cells which have low metabolic activity have lower number of mitochondria in them. This is also very important facts to remember. Please remember these all facts because so many MCQs have been asked in previous past paper of PPSC and FPSC exams from this topic. So please be attentive and focus. Now, chloroplast. Pigment containing organelles are plastid and present only in plants. Pigments means color containing organelles which have some color which are present only in plants are called plastids. Green pigment containing membrane bound plastids are called chloroplast, which having green pigment are called chloroplast. The green pigment is chlorophyll, which is an organic compound. The green pigment present in plastid is chlorophyll, which is an organic compound. Chlorophyll molecule resemble heme molecule of hemoglobin. Chlorophyll has magnesium ion while heme has iron ion in center. This is the difference between chlorophyll and heme ion. Diameter of chloroplast range about 4 to 6 micrometer. There are three structures in chloroplast: envelope, stroma and thylakoid. Please remember these three structures which are envelope, stroma and thylakoid. Most of the volume is covered by stroma which has circular DNA and ribosomes. Carbon dioxide is fixed in stroma to manufacture sugar and proteins are synthesized here. Thylakoid are flattened sacs or vesicles which arrange to form grana. Please remember very important facts how grana are formed. Grana are formed by arrangement of flattened sacs or vesicles which are thylakoid. There are 50 or more thylakoids stacked in a single granum. Single granum and plural grana. 50 or more thylakoid sacs are stacked together to form granum. Chlorophyll molecules are arranged on thylakoid, that's why it is green. Each granum is interconnected with other through non-green intergranum. The non-green part of chlorophyll is called intergranum or Sunlight is trapped on membranes of grana, which is thylakoid, and ATP is formed. How ATP is formed? When sunlight trap on the membranes of grana, then ATP is formed. Chloroplasts are self-replicating bodies like mitochondria. Mitochondria can also self-replicate, so chloroplasts are also self-replicating bodies, means they can form copies of their own. All colors except green are due to chromoplast in petals, flowers, means the color except green, any color except green, are due to chromoplast or called as chromoplast, which may be in petals, in flowers. Chromoplast help in pollination and seed dispersal. This is the main important function of chromoplast, which is pollination and seed dispersal. Now leucoplast. Leucoplasts are colorless and have triangular shaped. This triangular shape is important to remember because this MCQs I have seen in the past paper that triangular shaped chloroplasts are leucoplasts, or tubular shaped, which are found in the underground parts and store food. They are found in the underground part and their function is to store food. Please keep in mind. Now, nucleus. Robert Brown. Remember this name. Robert Brown reported nucleus for the first time in 1831 completely. It controls life and activities of cells. Usually cells are mononucleate, but they can be dinucleate or multinucleate, may have two nucleus or many nucleus. Nucleus in animal cell is in center, while in plants it is pushed near periphery or the outer boundary due to large vacuole. Nucleus can only be seen when a cell is not dividing because during cell division the nuclear membrane ruptures or disappears and chromosomes are in the cytoplasm. Inside the nucleus soluble sap is called nucleoplasm, which contain chromatin. Hereditary material or the genetic material is in the form of chromosomes when the cell is dividing. Chemically, nucleus have DNA, RNA and proteins. Nuclear membrane has pore for exchange of material. 30,000 pores in egg cells while 3 to 4 pores in erythrocytes. This is very important fact to remember that in egg cell there are 30,000 pores while in erythrocyte 3 to 4 pores are present. Nucleolus, and other important organelle in the nucleus. Organelle may also called structure. Darkly stained body in the nucleus without membrane is nucleolus. Please remember that the body without membrane is nucleolus. Ribosomal RNA is synthesized and stored in nucleus. Which function is performed by nucleolus? Ribosomal RNA is synthesized. Nucleus has two regions. The first one is peripheral granular area and central fibrillar area. Granular area has ribosomal subunit precursor and the fibrillar area has large molecular weight RNA and ribosomal DNA. Ribosomes are assembled in nucleus and then exported out. Now, chromosomes. Nucleus is deeply stained with basic dyes due to chromatin. Darkly stained thread-like structure in nucleus are chromosomes. Chromosomes have arms and centromere. Centromere is the place where spindle fibers attach. Centromere is the central part. Chromatid is the exact replica of chromosome. Chemically, chromosomes are made up of DNA and proteins. Genes are present on chromosomes, which are unit of inheritance. Please remember that human has 46 chromosomes, frog has 26, chimp has 48, fruit fly has 8, onion has 16, potato has 48 and pea has 14 chromosomes. Germ cells have haploid half number of chromosomes. These are very important MCQs in the past papers. Prokaryotic versus eukaryotic cell. Eukaryotes have prominent nucleus with nuclear envelope, while prokaryotes don't have any nucleus. Bacteria and blue-green algae are prokaryotes. Prokaryotes lack membrane-bound structures like mitochondria, endoplasmic reticulum, chloroplast, Golgi apparatus, etc. Ribosomes of prokaryote are small, which are of 70s, while eukaryote are 80s. Mitosis is missing in prokaryotes and they use binary fission. Most distinct feature of prokaryote is cell wall made from polysaccharides and amino acid to form murine or peptidoglycan. Their cell wall is considered as a single molecule called as sucrose. That was chapter 4, and the upcoming video will inshallah complete chapter 5, complete concept and summary. If this video is useful for you, please like, share and subscribe my channel. Stay connected with me. Allah Hafiz.